My man.
Hello.
Yeah, what up?
Nothing much. Let me get ready. Are there any questions before we begin? Uh, so we're going to be talking about Lab 00. Let me briefly go over the syllabus. I think that's it. And where's the other one? Oh, come on. I got the syllabus right here. So let me share my screen. I am recording. Is there anyone here in the lab who is not registered for the course and who would like to add the course besides Sheba Bali? Oh, come on. I guess I have to stop sharing my screen to get that information. So everyone here is already enrolled in the course. If you're not, you need to let me know. Oh, Shiva Bali is here. Just unmute the screen, and how you do that is you click on the bottom left of the Zoom screen, and there's a mute and unmute button there. So everybody else is enrolled in the class. I just need to know. So it looks like there's only Shiva trying to get in. All right, let me close that. I don't know how to do that. Share my screen, maybe that'll close it. Yep, I did. All right. So we're going to talk about Lab 00, lab safety training. You must turn in the worksheet to this lab. Why can't I get that up higher? There we go. You must turn in the worksheet by 8 a.m. on Friday for Lab 00. Remember to take the online plagiarism quiz by this Saturday, and you must receive 100% in order to get full credit on your infectious disease project. All right, let me see. I'm not having control of the mouse for some reason. There's just a few parts I want to talk about. I've already talked about the textbooks. The lab will be a part of this class. Microscope use in labs and virtual labs. You're not going to be using a microscope because everything we're going to do is online, including the labs. However, we will discuss microscopes in the lab in a future lab, and then you will need to know about microscopes. We do have some virtual labs, and any virtual lab that does not use Flash, you will be required to complete. We do have a virtual lab on the microscope where you will be using a computer program that'll be simulating using the microscope. But it too will be online. I've heard 2 people add the class lately. Just let me know if there's anyone who's attending who would like to add the class. I need to know who is attending if you want to add the class. All right, let's talk about safety information for students and physicians. The following is included for educational purposes as future nurses, dental hygienists, and other healthcare workers. It's important of you to know and be aware of these issues. In an online class, you're not going to be infected with any microorganisms because we will not be working with an actual microorganism. But you must complete page 4 of this document on Canvas website. No, you don't need to complete that. But the complete version of this is online. It's called the Document Information for Students and Physicians. And we have a copy of it in the physical lab. What you would want to do if you were actually going into a real microbiology lab and be exposed to microorganisms is to monitor yourself to see if you ever become sick. All of the organisms used in our microorganism are fairly safe organisms, but we operate the lab under biosafety level 2, where we have personal protective equipment like lab coats, gloves, goggles, etc., that you are required to work with in the lab. And then the lab is restricted to who can have access to it, as well as what infectious agents are in the lab. I'll let you read about students who are immune or compromised, or living or caring for someone who's immune compromised, because you're not really going to need to work with microorganisms. But we have down here a list of the species that are grown every term in the lab. Students only handle BSL-1 experiments and organisms. Those are biosafety level 1, but in the lab, we do have some biosafety level 2 organisms which present medium risk. And here are all the organisms in the lab. Let's talk about the lab and safety rules. Laboratory safety rules are a very important part of microbiology. Although we're not going to be physically meeting in a campus microbiology lab, you do need to learn about our lab safety protocols as part of this course. Many nursing programs also report that safety is one of the main skills that they want their students to learn. Safety is of primary importance in this laboratory. The attitudes and actions of persons working in the lab determine the level of safety. So always follow the procedures as they are given in the lab. They are to reduce the risk of microorganisms contaminating you, your lab space, and then the other people in the lab. So develop a positive and respectful attitude towards microorganisms. Never take shortcuts where risk and safety are shorted, meaning don't take shortcuts. You're not allowed to take any personal items to your lab bench or into the lab proper. Everything that you bring into the lab, such as your bag, your books, your purse, have to be put in the cubby holes, which are not considered inside the lab. So when you walk into the lab, the door and the cubby holes where you store items are not considered inside the microbiology lab. They are considered outside. And then when you walk further into the lab, that is the actual inside the microbiology lab. Only enrolled microbiology students are allowed into the lab unless authorized by the instructor. All personal items should be put in the cubby holes, which include book bags, books, purses, cell phones, and coats. anything that is brought into the lab will be confiscated and then sterilized before you can take it home. How it will be sterilized, it will be put in a bucket of bleach for 15 minutes, or it would be autoclaved. So if you were to take your cell phone into the microbiology lab, it would be confiscated and then sterilized, and either treatment would destroy your cell phone. You could take in a paper and pencil or a pen, as long as you leave those items in the lab for eternity. You could not get any of those items back until they have been sterilized. Now, a pencil and paper can be autoclaved, although autoclave will make the reading a little difficult on the paper, but it can be autoclaved. And then a pen, probably best for a pen to be stuck in the bleach, and then you can take it out of the lab. Any question about any of that? The only personal items you can bring into the lab are something you're wearing, and you should not touch it while you're in the lab. So if you have jewelry on, you can bring that into the lab. If you have your eyeglasses on, you can bring it into the lab. Now, the difficult part with the eyeglasses is you should remember to never touch them. And hopefully, if they start falling off, you would sterilize your gloves before you touch your glasses. You can also take in a watch. So those are a few personal items that can be taken in. Anything that you're wearing essentially can be taken in to the lab. And then you should not touch them once you're in the lab, because if you touch them, you can transfer microorganisms to them. All right, all cultures you will be working with are potentially pathogenic, so keep hands, pencils, and other materials out of your mouth, ears, nose, etc. There's no eating or drinking or smoking or gum chewing or any other oral habit allowed in the lab. When you're in the lab, you're always going to have nitrile gloves on. Their use is required, at least when you're working with microbes. Obviously, when you're coming into the lab and you're leaving the lab, you can take the gloves off. These gloves are supplied for you. You don't need to bring them. You must wear proper attire in the microbiology lab. No open-toed shoes are allowed in the microbiology lab. And that would be something like a thong or a sandal or a woman's fancy shoe that is open-toed. So you should only have closed-toed shoes in the microbiology lab. Why? Because if an accident happens and you spill a culture on your feet, we want minimal protection for your feet so that the microbes will not get on your feet. And tennis shoes give only minimal protection, but they do give some protection. And so tennis shoes are allowed, but closed-toed shoes must be worn. You may wear open-toed shoes and then bring a pair of closed-toed shoes and put them on in the lab. That would be kind of a waste of time. What you'd really want to do for any day you're going to go into the lab, you should just put on closed-toed shoes for that day. Lab coats are required in the lab. They are to be worn completely buttoned whenever you're in the lab proper. And lab coats can either be purchased in the bookstore and then brought into the lab, or we have many lab coats which are supplied by us in the lab. The lab coats will be kept in the lab for all the term, and so you will always use the same lab coat each lab. And if you brought it in, you can never take it home until after the end of the lab, in which case the lab coat would be autoclaved and then washed and then dried and then hung up for you to come and pick it up and then take it home. Any questions about any of that? Goggles are required anytime in the lab when you're working with a liquid culture, a broth, or when a splash hazard exists. Like when you're working with the gram stain, some of the reagents would be hazardous if they splash into your eye, and so you must wear goggles. The lab is equipped with safety goggles for your use, and they are disinfected with UV light. So we supply the goggles, but you must wear goggles whenever you're working with a liquid culture or with a splash hazard. Cleanliness is a virtue in microbiology. So before you begin each day in microbiology, you should disinfect your lab bench beforehand. And then after you're done at the end of the day, before you leave, you should also disinfect your lab bench. You put down disinfectant and then take some paper towels and spread it around and then wipe it off. When you come into the lab every day, you should put your things in the cubbyhole and then walk into the lab proper, wash your hands at the sink, then go and put on your lab coat, then go and get the gloves and put on the gloves, and then go to your lab station in the lab proper. If you're going to be working with a splash hazard, you may at this time go and get the goggles. Any question about any of that? And then when you leave for the day, you would wipe down the lab bench and then take off the gloves. Well, first take off the goggles if you have goggles, put them away. Then take off the gloves. Then take off your lab coat, hang your lab coat up, go and wash your hands, and then you can go to the cubbyhole and grab your things and leave. And so that procedure is used each time you come into the lab and each time you leave the lab. Accidents should be reported to an instructor. Minor accidents should be cleaned as follows. Always clean broken glass first. Broken glass is a hazard and can cut your skin from the broken glass, especially if it's contaminated with microorganisms. So use a brush and a broom to pick up the glass. Never pick it up with your fingers. And dispose of the broken glass in the proper receptacle. We have a receptacle for putting broken glass. If it's contaminated, before you pick up the broken glass, you should spray it with disinfectant and then cover it with paper towels so that will wick the disinfectant everywhere, and let that sit for 15 minutes. And then pick up the broken glass. If you spill a strong acid or base, they should be diluted and then wiped up. Spilled bacterial cultures should be cleaned the follow. Cover the spilled material with lots of paper towels. Drench the paper towels with disinfectant. Let the disinfectant stand 15 minutes before wiping it up, and then wipe it up and wipe it up with paper towels to wipe up the liquid. If there's any broken glass, remember to pick that up first and then put that in the broken glass container. All of the other material, like the paper towels that have bacteria on them, should be discarded in the biohazard trash can. You should keep your hair and paper away from the Bunsen burners and the Bacto incinerators. If you have long hair, it should be tied back before coming into class or immediately once you enter the lab. Whenever you're not using the Bunsen burners or the Bacto incinerators, they should be turned off, and they should be turned off before you leave the lab. Mouth pipetting is never allowed in the microbiology lab. Never take cultures out of the labs. Cultures should only be worked with in the lab and stay in the lab. Samples may be brought into the lab only when authorized by an instructor, and you can request that if you wanted to bring in some sample and then do something with it in the microbiology lab. But you need to get clear it with me first. If you have a cut or a sore on your hand, you should wash your hands as normal, apply a bandage, and we do have Band-Aids in the first aid kit in the lab, and then put the glove on over the Band-Aid. And then when you leave the lab, take off the glove, go wash your hand, and it's up to you if you want to replace the Band-Aid or not. All microbial cultures in the lab must be completely labeled with your name, the date, the nature of the specimen, like for example, if it's E. coli, write E. coli, and then the medium used. So if it's tryptic soy agar, put TSA for tryptic soy agar. Old specimen must be discarded properly in the following ways. Agar plates should be discarded in the appropriate biohazard container. Do not put agar plates with bacteria in the trash can. Glass slides and cotton swabs should be put in the appropriate container containing disinfectant, and they're found at the rear of the lab. And the swabs, we have containers on each lab bench or each lab table as well. Pipettes should be put in the appropriately labeled container. If they are a glass, put them in the appropriately labeled container for pipettes. If they're plastic and they do not have bacteria on them, they can be placed in the trash can. But if they're glass, never put glass in the trash can. And that's true of a pipette or any other thing that's glass. Never put glass in the trash can. For that matter, never put glass in the biohazard container either. Like the broken glass should be put in the broken glass container. Pipettes that have bacteria on them that are glass should be put in the pipette container. Slides should be put in the slide container. We do have a bucket for test tubes, not a bucket, a test tube rack for putting used test tubes in, and those are in the back of the lab. So put them when you're done in the test tube rack. The test tubes and the slides will be taken from the lab, will be autoclaved, will be washed, and then will be made available for later use. Any question about any of that? I'll let you read about supplies for the lab because we're not going to really be in the lab. And you can read about the drawers too because we're not really going to be in the lab. So anything that you use in the lab, like paper and plastic that do not have bacteria in them, go in the trash can. If they do have bacteria in them and they're paper or plastic, they go in the biohazard container. If they're glass, they go in the appropriate container. Never put glass in the trash can or in the biohazard container. You are required to follow all of these rules, and you will be tested on these rules in quiz number 1. You might be tested in some of these rules also in the practice quiz. Any questions about any of these rules? All right. So today we're going to be talking about Lab 00, lab safety training. Remember to turn in the worksheet to this lab by 8 a.m. Friday. you should read the infectious disease research project. It's called the project instructions on Canvas. Let me show it to you. So read the infectious disease research project for next week. I'll be talking about this project next week. And this project's worth 100 points. Any question about any of that? Any question about what we're doing? All right, if not, let's go to the lab. Let me go ahead and go to the time here. We have a lab module and also a lab worksheet. I guess I should show that. So there's the lab module for lab 00. And then the worksheet, click on this link here. Oops, I shouldn't have done that. And then click on that link there to get lab worksheet for lab 00. Here is the lab module here. Laboratory safety, lab module 00. Online students are not going to be working in the lab, but you do need to know this material, as many of these rules will be relevant during your career. The primary learning objectives are: 1, be able to distinguish between safe and unsafe laboratory practices, understand where relevant safety equipment is located in the lab, understand how to safely dispose of contaminated materials. Is this the worksheet? All right, here we go. I'm not seeing my highlighting here, so I'll have to wing it. Apparently, I don't have that in the summer for some reason. Microbiologists are expected to observe a number of safety rules, as they will be working with potentially pathogenic microorganisms. A pathogen is an organism that can cause human disease. While you're not actually going to be performing any of these laboratory things, it is important that you understand the guidelines required to create safe laboratory environment. Safety precautions include wearing proper attire. This includes a clean lab coat that covers your lap when sitting, disposable gloves, closed-toed shoes, goggles when working with a splash hazard, and a few other things that are not listed here, such as, well, closed-toed shoes is listed here, but you have to be wearing pants, you have to be wearing a shirt or a blouse. If you have long hair, the long hair should be tied back, and you should have a hair tie on. Lab coats, goggles, and gloves must be removed before leaving the lab. They will be remaining in the lab for the entire duration of the lab for the entire quarter. Hands must be washed thoroughly with soap and water before entering the lab and before leaving the lab. Absolutely no food or drink in the lab at any time, or any use of electronics. No personal items are allowed into the main part of the lab, including cell phones. There is an exception here, and that is if you're wearing the personal item, you can bring it into the lab. Like if you have jewelry on, you can bring that into the lab. If you have a wristwatch on, you can bring that into the lab. You should not touch the personal items while you are in the lab. All other personal items have to be stored in the personal storage area, the cubby holes, by the door, and we do consider that part of the lab outside of the lab. It's actually inside the door, but the door and the cubby holes are not considered a part of the microbiology lab. they are considered outside the microbiology lab. Any personal item that is brought into the main part of the lab will be confiscated and not returned to the student until they've been sterilized by autoclaving or soaking in bleach for 15 minutes. And that will destroy any electronics. Here is what a person should look like when they come into a lab. Closed-toed shoes, pants, a lab coat, gloves. The lab coat's completely buttoned. This person looks like they either have their hair tied back or they're wearing a hair net. And that's not goggles the person is wearing. Those are actually their personal glasses. If you do wear glasses, you are allowed to bring the glasses into the lab. The difficult part is to remember to not touch your glasses. If you do need to touch your glass, you need to sterilize your hands, and you could put like alcohol or disinfectant on your gloves, rinse them, and then dry your gloves and then touch the glasses. No food, drinks, cell phones, headphones, etc. are brought into the lab. Keep your work area clean and uncluttered. I mentioned this when I was talking about the syllabus. You should disinfect the lab station where you work at the beginning of each lab session, as well as at the end of each lab session. Here is an actual picture of our lab showing the place a student would work, and then that's either a sink over here or another space where somebody can work. Discard waste in the appropriate receptacles. Regular trash that has no contamination on it and is not sharp, meaning not glass. We don't really use other sharps like a scalpel or a needle, but those are not allowed into the regular trash. But paper towels and gloves that are do not have bacteria on them can be put in the trash can. Anything that has bacteria on it, and that includes contaminated paper, contaminated gloves, agar plates, should be put in the biohazard container. No glass ever goes in the biohazard container. So no slides, no beakers, no test tubes, no glass pipettes either. We do have bins or a rack for putting contaminated test tubes. When you're done with them, put them in the test tube holder, and that will be at the back of the lab. For swabs, slides, and cover slips, we have a separate container for each of them. They all have disinfectant in them, and the swabs will be put in the disinfectant, and then I think they're thrown away after they've sat in disinfectant for 15 minutes. The cover slips we do not reuse, so they would be the disinfectant would be poured off, and then the cover slips would probably be disposed of as broken glass. And then the slides will be, they've already been disinfectant, so they would be washed and then reused. If you're not sure where to dispose of something, ask your instructor. Report all spills and accidents immediately, no matter how slight. Cover the microbiology spills with paper towel, and then saturate the region with disinfectant, allowing the disinfectant to soak for 15 minutes before you wipe it up. And that could cause a problem on the floor. Just don't walk on that area, and you see that on the floor, just avoid that area until it's picked up. Keep lids and reagent bottles on when you're not using the reagents. Never pour reagents back into containers. Do not pour chemicals down the drain. We do have regions to pour the chemicals in liquid biological waste, or liquid, not biological waste, liquid chemical waste. and that is disposed of appropriately. I'm not sure what they do with it, but they will get a large vat of it and then call somebody up to come and pick it up, and it's treated as a biohazard. So it's not poured down the drain, and it's not buried with the normal trash. I'm not sure what they do with it. They probably either incinerate it, or they put it in a special buried site with other hazardous material. No mouth pipetting, no putting things in your mouth, including fingers. Of course, you should have gloves on. Nobody would ever put gloves in their mouth. Oops. When Bunsen burners and Bechtel incinerators are not turned on, when they're not being used, don't turn them on, turn them off. Bunsen burner shown here, and a Bechtel incinerator. Both of them, what you want to do is sterilize the wire portion of the instrument so it becomes red hot. You don't sterilize the handle portion, otherwise you wouldn't be able to handle it, but you do get red hot the wire portion, and that's what we do to sterilize instruments we use in microbiology, such as a loop. That's a loop there, and that's probably a loop there. It's hard to tell because I can't see it. You should become familiar with the location of safety equipment, and because you're never going to be in the real lab, you don't need to know where the safety equipment is, but you should know in each microbiology lab, we have an eye wash station shown here. We have a deluge shower shown here, and hopefully I'll never have a student use this because that sprays water all over the floor, meaning there's no place for it to go except on the floor. And we do have a drain on the floor, but still, if you ever spill anything on you, like a hazardous chemical, and you want to go into the deluge shower, we have one for you, but hopefully we'll never use it. I've never used it in all the years I've been teaching, and I've been teaching since 2003. You should know we have a fire alarm, we have a first aid kit, we have a fire blanket, which is, I think, what that is. And then a fire extinguisher. That is safety equipment. So when it asks what the safety equipment is, it's listed here. Don't put something like a lab coat. That is not a piece of equipment. That's a piece of clothing you wear for safety, but it is not a safety equipment. Did I show everything? Yep, there we go. I did show you the fire blanket. And you know, I don't know what that one's supposed to be, because that's the fire alarm. That looks like another alarm, but I don't know what it is. It's not the fire alarm. It must be some other alarm. So whenever in doubt, always ask. Now complete the laboratory exercise in a separate file. Do not put in the material on the lab module here. This is the same stuff I read to you in the syllabus. So I'm not going to go through that again. Just make sure I covered everything. Never take cultures out of the lab. I think I did mention that. All cultures should stay in the lab and never be brought out. I mean, you have to get authorization if you want to bring in a culture to the lab. Everything you have in the microbiology lab should be completely labeled with your name, the date, the nature of the specimen, what it is, like E. coli, and then the medium used. All right, that's it. And for the lab, let's go to the worksheet. Answer the questions in the worksheet and then turn it in and you're done with the lab. This part of the lab doesn't work well, and it doesn't work unless you print it out. And I'm just going to tell you, skip the first page. It does have some questions for you, but just skip the first page. I've got the questions on the second page, and I've got ignore the above. Here's the lab safety word scramble. So fill in each asterisk. So like in rule 1, you have to give 4 answers because I have 4 asterisks. In the lab, what do you always wear for safety before you enter the lab? So you have to put this on before you enter the lab. And you should put it on before you enter the door to enter the lab. Okay? In rule 2, after you enter the lab, what do you put on to keep yourself safe in the lab? So anything in rule 2 should not be in rule 1, and anything in rule 1 should not be in rule 2. Because rule 1, you're putting it on before you enter the microbiology lab. In rule 2, you're putting it on after you enter the microbiology lab. Any questions about any of that? All right. Students always seem to get that confused, and I'm trying to straighten that out. In rule 3, you need to look up online what is a safety data sheet. A safety data sheet describes a chemical that's present in the lab, and we must have a safety data sheet for each chemical in the lab, and the safety data sheet describes, list 1, 2, 3, 4, 5 items the safety data sheet describes. And by the way, this is not a single word answer. To answer this, you must answer with at least a phrase. You may answer with a sentence, but you need at least a phrase to answer this. If you just put in time, I will say, what do you mean by time? Explain that. Okay? All right. Rule 4, know the location of your safety equipment that is in the lab, such as, and then there's 1, 2, 3, 4, 5 different safety equipment that you have to name. Rule 5, what can you take into the lab? And don't say nothing, because there are a few things you can take into the lab. Obviously, you can take yourself, okay? And you can take the clothes that you're wearing, but the idea is there's a few other things you can take into the lab. And then, can you eat and drink in the lab? And once again, you have 3 different things to answer here. And question 2, before those were question 1, rule 1, 2, 3, 4, and 5, to question 1. And then 2, list 1 item in the laboratory attire that you must wear in a microbiology lab. And then 3, list 1 item that is allowed in the microbiology lab. So you don't have to have it, but you may take it into the microbiology lab. And then 4, list 1 item that is not allowed in a microbiology lab. And then submit this file by 8 a.m. on Friday in the folder for worksheet lab 00. Only submit the worksheet. If ever you submit the lab module, the first time I will give you a warning. The second time you submit the lab module, you will get a penalty. Why? Because the lab module is much bigger than the worksheet. And if everybody were to submit lab modules, my Canvas website would fill up. Once it fills up, students will no longer be able to turn in assignments. I do not want that to happen. So please only turn in the worksheet, never turn in the lab module. Any question about any of that? All right, if not, go ahead and work on the
I have
lab.
a question.
Yeah, we're done.
Oh, I have a question. For rule 1 and rule 2, that's kind of confusing because to me it looks like the answer would be the same.
No.
Okay,
because Rule 2, you're putting on something after you enter the lab. Rule 1, you're putting on before you enter the lab. Most people will put on rule 1 in their own home,
okay, I get it now. Thank you.
right?
Thank you.
Right. All right, I'll be here until 9 o'clock. I doubt anyone will be here until that late, but I will, let's stop the share here, stop recording. I'll